Hello friends, welcome to this video where we are going to discuss cell organelles, a very simple basic topic which you have done in your school time. But by making this video, I just want to make sure that you have revised this simple topic. So let's start. Cell organelles. Let's see cell. Cell is the structural and functional unit of life. We have eukaryotic cell and prokaryotic cell. Let's quickly see the differences between prokaryotic and eukaryotic cell. Pro means primitive or before. Karyo means nucleus. So this is a primitive and simpler cell which was existing before the formation of nucleus, right? Because there is no proper nucleus in this prokaryotic cell. On the other hand, eukaryotic cell is a well developed cell with all the organelles which are membrane bound organelles, and there are no organelles in prokaryote, right? Now let's see cell wall and plasma membrane. Cell wall is present in prokaryotes, but in eukaryotes it is only present in eukaryote plant and fungi. It is not present in human cell. One more thing in cell wall, that cell wall is chemically complex in case of prokaryotes, but cell wall is simpler in case of these eukaryotes where it is present. Then cell membrane. Cell membrane is present in eukaryotes also, present in prokaryotes also, but in eukaryotes there are receptors attached to cell membrane, but these receptors are not present in prokaryotes, right? Then next point is regarding nucleus and the genetic material. In case of prokaryotes, the portion of cytoplasm where genetic material is present is known as nucleoid. So we say that there is nucleoid in prokaryotes, but not a proper membrane bound nucleus. And the genetic material is circular double stranded DNA. And as there is no nucleus, so transcription and translation both occurs in cytoplasm, and they can occur simultaneously. On the other hand, in case of eukaryotes, there is a proper membrane bound nucleus with nucleolus also present in it, which is rich in rRNA. And here the DNA or genetic material is linear double stranded DNA. In this case, transcription, that is formation of RNA, occurs in nucleus, but translation, that is formation of proteins, that occurs in cytoplasm, right? Then next point is ribosomes. Ribosomes are smaller in case of prokaryotes, that is 70S ribosomes are present in prokaryotes, but larger ribosomes are present in eukaryotes, that is 80S, right? So you can see all this in the picture given. So cell membrane present in prokaryote also, eukaryote also, cell wall present in prokaryotes, also in some eukaryotes. Then nucleoid is present in prokaryote, ribosomes small in prokaryotes, large in eukaryotes. Mitochondria or any organelle will be only present in eukaryote. Then membrane enclosed proper nucleus with nucleolus is present in eukaryote. Now let's see a clear bigger image of a eukaryotic cell which is complex and having so many organelles in it. So this is mitochondria, this is plasma membrane, then microtubules are present in eukaryotes, centrosome, microfilament, then lysosome is an organelle which is present, endoplasmic reticulum, smooth which is not having any ribosome attached to it, rough endoplasmic reticulum which is having ribosomes attached to it, and this is nucleus, this is nucleolus, this is nuclear membrane, this is nuclear membrane, and you can see the endoplasmic reticulum sacs are extension of nuclear membrane. Same color shown here in the picture. You can see blue color nuclear envelope that is only getting continued into the sacs of endoplasmic reticulum here, like this. Okay, so very easy to visualize in this picture. Then Golgi apparatus, also membranous sacs present here in the cytoplasm. Then peroxisomes are present, peroxisomes organelles, which helps in the degradation of free radicals. Then ribosomes, which are not considered organelles. Ribosomes are not considered organelles as they do not have a membrane. Organelle means one which is having a membrane, right? So let's take the organelles one by one. Mainly there are 6 organelles in a eukaryotic cell. The largest organelle is nucleus, having the genetic material, then mitochondria, the energy house of the cell making ATP, then peroxisomes for free radical destruction, then lysosomes, then endoplasmic reticulum, then Golgi apparatus. So let's take them one by one. First I will be taking nucleus. So nucleus having nuclear pores. Nucleus has 2 membranes. Nucleus has 2 membranes, inner membrane and outer membrane. So at places inner and outer membrane is joined. So this is inner nuclear membrane, this is outer nuclear membrane. At places inner and outer is joined to make nuclear pores which are big in size, because bigger substances have to leave the nucleus and enter the nucleus. For example, ribosomes, DNA polymerase, RNA polymerase. These big things are getting transferred from the nuclear pores, so they need to be large in size. Then the eukaryotic cell has proper condensed chromatin, which is having DNA as well as histone proteins attached to it. Then there is a nucleolus, which is rich in rRNA, helps in the formation of ribosomes, and it disappears during cell division, right? So done with this organelle, nucleus. One more thing about inner mitochondrial membrane is that how this membrane is made semi-permeable. This is because of the presence of a special phospholipid, that is cardiolipin. This cardiolipin is a special phospholipid which has 4 fatty acids in it, so it is more non-polar also. Instead, normal phospholipid has 2 fatty acids in the structure, right? So due to the presence of cardiolipin, the membrane is made semi-permeable. Then, regarding mitochondria, these are self-replicating organelles, as mitochondria have their own DNA, their own ribosomes, and the protein synthesizing machinery. So they synthesize few of the proteins of mitochondria also, and they synthesize 20% proteins of etc also, right? Then there are multiple mitochondria per cell. Usually students think that there is only one mitochondria per cell, but no, they are variable in number, they are multiple in number usually, and it depends on how much energy is required by the cell. So if more energy is required, then more mitochondria will be formed there. So with this, we're done with mitochondria. Coming to the next organelle, that is peroxisomes. Peroxisomes have a single membrane. Like we did that nucleus has inner and outer nuclear membrane, mitochondria has inner and outer membrane, but peroxisomes have only a single membrane. In peroxisomes, whatever pathway occur, ATP formation will not occur in that pathway. Instead, there is formation of H2O2 in most of the pathways occurring in peroxisomes. So, as this free radical H2O2 is regularly getting formed in peroxisomes, so it needs to be regularly destroyed also. Therefore, peroxisomes have enzymes like catalase, peroxidase for the destruction of this regularly forming H2O2. Now, which pathways occur in peroxisomes? Alpha oxidation of fatty acids occur here. Oxidation of very long chain fatty acids occur here, and synthesis of plasmalogen occurs here, which is the main phospholipid of myelin in the brain. That's why any disorder in peroxisomes is going to affect CNS, right? Then peroxisomes are also given another name, that is glyoxysomes. Also they are called microbodies. Then disorders in peroxisomes. There is a rare group of disorders known as peroxisomal biogenesis disorders, PBD. Peroxisomal biogenesis disorder. All these disorders will be affecting CNS, I already told you. And the most severe peroxisomal biogenesis disorder is Zellweger syndrome. This detail of this topic I have covered in lipids, so you can do it there, and it is very, very important and frequently asked topic in the exams. Basically, in all the peroxisomal biogenesis disorder, what is happening? There is problem in the synthesis. See the word biogenesis, synthesis of peroxisomes, right? Okay, so done with peroxisomes. Next, coming to lysosomes. Lysosomes, also known as digestive organelle of the cell. What digestion is required in the cell? Digestion or breakdown of macromolecules is required. Macromolecules. Like body's digestive part is intestines. Similarly, in a cell, the digestive organelle is lysosomes. Whenever it's time for some macromolecule to get broken down, it will move towards lysosomes and will be broken down. So this is the digestive organelle. And then which enzymes will be present in lysosomes? Hydrolases, hydrolytic enzymes, because hydrolases will break down macromolecules, and The optimal pH here is approximately 5. Acidic pH. These enzymes have acidic pH and they break down macromolecules and they are normally present in the lysosomes. So, a normal lysosome having these hydrolase enzyme is called a primary lysosome. It will get fused with the vesicle containing the macromolecule, or it is containing some microbe. Then these two things fuse to make secondary lysosome, where the enzymes of lysosomes will digest the toxic material. Now, the diseases in lysosomes are known as lysosomal storage diseases. Lysosomal storage diseases. What do you mean by lysosomal storage disease? I told you molecules gets broken down in lysosomes by help of hydrolase enzymes. So if some hydrolase enzyme is not there, the particular substrate macromolecule will be raised and will be stored in lysosomes. That disease will be called lysosomal storage diseases. So let's list all the lysosomal storage diseases. Number 1, MPS, mucopolysaccharidosis, where mucopolysaccharides get accumulated. Detail of this topic I have taken in carbohydrate chemistry portion. Then eye cell disease or inclusion body disease I have taken along with MPS in carbohydrate chemistry. Then one of the glycogen storage disease, that is Pompey's disease, which is type 2 glycogen storage disease. Then SLP, that is sphingolipidosis. I've covered this in lipid chemistry portion. In sphingolipidosis, there is Gaucher's disease, which is most common lysosomal storage disease. Then Wolman's disease is covered along with sphingolipidosis. Wolman's disease also is a disease in lipid chemistry portion only, but Wolman's disease is not a sphingolipidosis. Be careful, I repeat, Wolman's disease is a lysosomal storage disease, but not a sphingolipidosis. And I can say that all the sphingolipidoses are lysosomal storage diseases, right? Then, sixth and last is cystinosis. Cystinosis is a lysosomal storage disease, and I've covered this in sulfur containing amino acid metabolism video, right? So I have listed all the lysosomal storage diseases here. Now let's also note down that which lysosomal storage diseases have enzyme replacement therapy available, ERT available. These are mucopolysaccharidosis type 1, 2 and 6. These have enzyme replacement therapy available. Then Pompey's disease, ERT is available. Then in sphingolipidosis, Gaucher's disease, Niemann-Pick disease and Fabry's disease, enzyme replacement therapy is available. Also in Wolman's disease, ERT is available, right? You can just note down, question can be asked. And with this, we are done with this organelle, lysosomes. There are two types of endoplasmic reticulum. Rough endoplasmic reticulum, that is where ribosomes are attached. Okay. Other one is smooth endoplasmic reticulum, where ribosomes are not attached. You can see in the picture, this is smooth endoplasmic reticulum, this is rough endoplasmic reticulum. These red dots, these small red dots are ribosomes attached here. What is the role of these two? The role of rough endoplasmic reticulum is in the synthesis of proteins. Which proteins? The proteins which are to be exported out of the cell. Example, hormone, insulin. So insulin will be synthesized on rough endoplasmic reticulum. On the other hand, smooth endoplasmic reticulum has no role in protein synthesis. Instead, it has role in three things. It has role in detoxification process, which mainly occurs in liver. It has role in lipid synthesis, example steroids, phospholipids, and it has role in calcium sequestration and release. Right now there's a term called sarcoplasmic reticulum. It is that smooth endoplasmic reticulum which is present in myocytes of smooth and striated muscles. Right? Okay, now what are microsomes? Earlier I told you microbodies. What I told you? That peroxisomes are also known as microbodies. Okay, but now I'm talking about microsomes. What are microsomes? These are disrupted endoplasmic reticulum vesicles which are formed in vitro. You know that in vivo endoplasmic reticulum is like membranous sacs, right? In vitro it can be disrupted and it gets converted to various vesicles. These are known as microsomes. So microsomes are not present in the cell in vivo, but they are just formed during the cell disruption. Okay, so with this we are done with this endoplasmic reticulum. Now the last organelle, that is Golgi apparatus. Golgi, G for Golgi, G for gift packing. This organelle receives the proteins from the rough endoplasmic reticulum and will do the packaging of those proteins which are to be released out of the cell. So, see the diagram. These are also membranous sacs. This side is receiving end of Golgi apparatus, which is receiving vesicles, protein containing vesicles from rough endoplasmic reticulum. So protein is getting synthesized in rough endoplasmic reticulum, then it comes to Golgi apparatus. Here the packaging, sorting is done and new vesicles are formed, and this is shipping side of Golgi apparatus, via which the vesicle formed is released outside of the cell by fusion with the plasma membrane. Let's see what all is done in the Golgi apparatus. Glycosylation can be done, packaging is done. Sometimes protein is stored for some time in the Golgi apparatus. Then phosphorylation is done. Then tagging, segregation, post-translational modification, all these things will occur in Golgi apparatus, right? Let me clear here about glycosylation. O-glycosylation occurs in Golgi apparatus, but if N-glycosylation is asked, then that occurs in endoplasmic reticulum, right? N-glycosylation occurs in endoplasmic reticulum. Now, let's see what are protein targeting disorders. Protein targeting disorders. I told you tagging is done in Golgi apparatus. So if there is a defect in Golgi apparatus, tagging is not done, then the synthesized protein, the protein is normal and normally synthesized, that protein will not reach the target site. So it is a protein targeting disorder, right? We can list down the protein targeting disorders. Eye cell disease is a protein targeting disorder. Primary hyperoxaluria, then familial hypercholesterolemia, then Zellweger syndrome, and cystic fibrosis. You can just note down that all these are protein targeting disorders, right? So we are done with 6 organelles. Now let's do ribosome. I told you ribosome is not considered organelle as it is not a membrane bound structure, right? Now these ribosomes, also known as free or cytosolic ribosomes, in order to differentiate them from the ribosomes which are attached at the endoplasmic reticulum, making that endoplasmic reticulum rough endoplasmic reticulum. Okay, that was different. We have done that. Now this one is free or cytosolic ribosomes, as they are present free in the cytoplasm. They are required for protein synthesis of those proteins which are required inside the cell mainly for cells metabolism, right? And ribosome is usually having a large subunit and a small subunit, right? Ribosomes are made up of rRNA and proteins. rRNA is more in amount as compared to proteins, so rRNA is considered as marker for ribosome. What do you mean by marker? Anything which is used to detect that organelle, that is the marker of that organelle. So if in an unknown organelle I detect lots of rRNA, means that is ribosome. Okay, so this was about free ribosomes. Now let's see how organelles are separated in lab, because normally all organelles are present inside the cell, right? So how will we separate all the organelles? And because we need to understand the organelles, we need to see them in microscope, and we need to further do the experiments. So how that process is done: first a cell is taken, then the cell membrane, its membrane is lysed. Cell membrane lysed mechanically or with the help of some chemical. When the cell membrane is lysed, then we get a homogenized mixture of the gel-like material of cytoplasm and the organelles suspended in it. So we get mixture of organelles. Now rotate it in a centrifugal tube. Because it's a mixture of organelles, now we rotate it in a centrifugal tube at slow speed first. So what will happen? First, heavier organelles like nucleus will be precipitated. Then we extract the nucleus which is precipitated, and the remaining mixture is again rotated at a centrifugal speed which is now little higher than the previous one. Now the lighter organelles will be precipitated, and similarly we can keep on doing this process. This process is known as sucrose density gradient centrifuge. Centrifuge because centrifugal force is used here. Density gradient because a different density of sucrose mixture is used here, right? So this was the technique. Now when we have collected all the organelles, then how to detect which organelle is which one? So for that we need to know the markers for various organelles. For example, what is the marker for plasma membrane? Sodium potassium ATPase or 5' nucleotidase or adenyl cyclase. Any one can be used as a marker for the cell membrane. Then for Golgi apparatus, it is galactosyl transferase enzyme, as this enzyme is only present in Golgi apparatus. Then for cytoplasm, it is lactate dehydrogenase, this enzyme of anaerobic glycolysis present only in cytoplasm. Then ribosomes, I already told you, rRNA is considered as marker for ribosome. Then glucose 6-phosphatase enzyme, which is present in endoplasmic reticulum, will act as marker for endoplasmic reticulum. Then catalase for peroxisomes. Then for nucleus, DNA or DNA polymerase can be used as marker. For lysosomes, it is acid phosphatase. For mitochondria, it can be succinate dehydrogenase or glutamate dehydrogenase. Right? So learn all these markers, question can be asked. And with this, we are done with the video of organelles. Thanks for watching and best wishes to you.